Molecular-based therapeutic technologies offer many significant advantages over traditional pharmaceuticals. These benefits include faster development, highly specific gene or protein targeting, greater tunability, and even permanent integration into patients' genomes. Such attributes have brought molecular therapeutics to the forefront in the development of treatments for challenging illnesses, including cancer, neurodegeneration, hereditary and infectious diseases, and immune system disorders. Drug delivery and targeting, genomic integration, safety, and efficacy can all be finely tuned by modifying the genetic code. Plasmids have become a key element in molecular-based therapies because of their ability to transfer and store genetic information, the ease of DNA manipulation and propagation, and their natural capacity to be taken up by a variety of cells. Plasmids can be directly administered to humans to drive therapeutic actions at the molecular level. However, plasmids can also be transferred into cells in vitro to reprogram them to produce recombinant viral vectors and proteins, which are harvested and used for treatment. As made evident by the commercial release of effective SARS-CoV-2 vaccines in the past year, plasmids can also be used to successfully manufacture therapeutic mRNA via in vitro transcription. Plasmid DNA is routinely extracted from E. coli, but regardless of whether the plasmid is used directly as the therapeutic agent or a precursor for manufacturing, there are stringent requirements to ensure safety and efficacy. Residual salts, proteins, RNA, endotoxins, plasmid concentration and quantity can all adversely affect downstream production, experimental outcomes, and the health of the patient. Traditional plasmid purification technologies utilize slow anion exchange columns that are driven by gravity and lengthy alcohol precipitation steps that require a lot of handling. These can take hours to complete and are prone to inconsistencies and low yields, making them inconvenient for the screening of large numbers of potential candidates, which creates a major bottleneck in the development of molecular-based therapeutics.